This is a summary video for Biology Paper 2 of AQA GCSE Combined Science. It gives an overview of the topics of homeostasis, genetics and evolution, and ecology, and it will allow you to do a last-minute cram on the night before the exam. If you're taking GCSE Biology, also known as Triple Science, then I do have a longer video that also contains all of the triple topics. Before we start on the biology, a few words of general advice. It's really important in all of your exams that you're writing in black pen because your exam paper is going to be scanned and other colours just don't scan as well. Make sure that you're staying inside the box on the exam paper and watch out for sneaky units that you might need to convert. When you're writing extended response questions, bullet points are allowed and in fact even encouraged because they help to stop you from waffling and make life easier for your examiner. If you meet a question that says evaluate, in order to get full marks, you have to write a conclusion, and you should also be comparing both sides of the argument. If you have to write a method for, say, one of the required practicals, it's important that it has a logical flow. It's a good idea to write your method in terms of bullet points or a numbered list. Don't forget that you are also being assessed on your working scientifically skills. So for any investigation, you should be able to identify the variables and you should be able to explain that using control variables makes it a valid experiment, also known as a fair test. If you repeat the same experiment and see the same pattern, then that is repeatable data. But if peer review takes place and other scientists repeat your experiment, or if you do the same experiment with a different method, but you still see that same pattern, then this is reproducible data. Resolution is the smallest difference in your measurements that you can identify. Computerized methods are accurate, rapid, and sensitive. And if you're asked to draw a graph of some data, don't forget the line of best fit, and don't forget that in GCSE Science, this may be a curve. Systematic errors are caused by equipment that is giving an incorrect reading, and it's always by the same amount. So if you know how much that amount is, you can then calibrate your data by adding or subtracting appropriately. Whereas random errors are caused by a lack of precision and just normal fluctuations in the environment. So the solution to this is to repeat the experiment multiple times, remove any anomalous data, and then calculate a mean. It's also important to note that 10% of all the marks in GCSE Biology are for mathematical skills, and these need to be at an appropriate level. So not just adding and dividing, but things like decimals, percentages, and standard form. So make sure you've had a little look through the list and that you're feeling confident with all of these as well. And now it's time for some actual biology. The first topic in GCSE Biology Paper 2 is homeostasis and response. Homeostasis is the regulation of the internal conditions of either a cell or a whole organism to maintain the optimum conditions for function in response to internal and external changes. What that means is that the environment all around you or all around a cell is changing all the time. So your body or that cell responds to that by making sure that inside the cell the conditions stay where they want them to. So temperature, water content, and other conditions need to stay the same. This is controlled by negative feedback loops. This is where your body or the cell makes enough of a hormone or enzyme until eventually it becomes excessive or too much, and then production is inhibited and it stops being made. 3 examples you should be aware of are blood glucose concentration, body temperature, and water levels. If you aren't taking triple biology, then blood glucose concentration is the one that you need significant detail for. The way that homeostasis works is that a type of cell called a receptor detects a stimulus in the environment. This could be something like a change in temperature or there being more light. Then a coordination centre, which could be the brain, the spinal cord, or the pancreas, takes that stimulus and organises a response to it. This coordination centre will pass some kind of signal to an effector, which could be a muscle or a gland. This effector will then actually do the response. There are nervous responses from your nervous system, and there are also chemical responses, called hormones, from your endocrine system. And there are some responses that involve both of these systems. You might be wondering why the human body needs these 2 systems. The endocrine system works using hormones which are released from glands. It's slower and produces longer lasting responses. Your endocrine system is responsible for things like you going through puberty, but also for homeostatic responses like a response to dehydration, where it needs to go on for quite a long time. Your nervous system uses nerves or neurons, and they transfer electrical impulses. These responses are very fast, but they're also very short-lived. They're useful for a situation like if you touch something hot and you need to snatch your hand away before you burn yourself, but you don't need an ongoing response after that. The nervous system is made up of receptors such as the retina cells in your eye or pressure sensors in your skin, and it passes information in the form of electrical impulses to the central nervous system, or CNS. This is a combination of your brain and your spinal cord. The CNS then coordinates the response of the effectors, so that could be muscles or it could also be glands. Between those nerves or neurons, there are little gaps called synapses. This allows one neuron to communicate with multiple others. When an electrical signal has travelled down a neuron and it needs to get to the next neuron, it has to cross that synapse. And it does that by releasing a chemical called a neurotransmitter, which can diffuse across the gap between the two neurons. And this will then trigger the next electrical impulse. When you think about drinking caffeine or drinking alcohol and how one seems to speed you up and allow you to move faster and think faster, and the other slows you down, often drugs like these are affecting the speed at which neurotransmitters are either released or detected or travel. A reflex reaction is a rapid automatic reaction. It doesn't involve the conscious thinking part of your brain, so you've never had to learn how to do it. And you don't think about it, you were just born with reflexes. Reflexes tend to all be about stopping harm happening to your body, so it could be something like a reflex response to you touching something very hot or to a bright light. You need to be able to describe how a reflex reaction works. This is likely to be accompanied by a diagram which you can interpret. Firstly, there's a stimulus. That's the thing that makes the reflex reaction happen. What has changed in the environment? So you could say that you have a stimulus, which is a change in temperature, and this is detected by a receptor in the skin. For light, it would be a receptor in the retina cells of the eye and so on. The receptor then generates a nervous impulse, so that's an electrical signal that passes along the sensory neuron to the relay neuron. You've got 3 types of neuron and you need to know what they are. The sensory neuron is connected to the receptor, which is the thing that's doing the sensing. Then the relay neuron is going to pass on the electrical impulse through your spinal cord, and then it's going to relay it onto the motor neuron. Motor is all about movement, so the motor neuron is the neuron that's responsible for passing the signal onto the effector, which is going to cause your body to actually respond. So the effector might be a muscle in your arm, which causes you to move your hand out of the way so that you don't get burned. The first required practical in Biology Paper 2 is about measuring reaction times to see how they're affected by a factor like caffeine or tiredness. A partner lines up a ruler with your thumb and forefinger so that the 0 is in line. Your partner then drops the ruler without warning, so you don't have an opportunity to anticipate them and give a false reading. You catch the ruler as quickly as possible, and we then measure how far the ruler has fallen. The distance travelled by the ruler before you catch it acts as a surrogate for your reaction time, which you can later convert using a conversion table. To increase the validity of the experiment, you should always use the same hand for the test, and the ruler should always be placed in the same position, with the 0 mark in line with the top of the hand. This test on itself is not the full investigation. We then need to repeat this, varying something to see how it impacts reaction time. This experiment could be carried out by the same group of people before and after drinking a caffeinated drink, or by different groups of people who've taken different levels of caffeine. If you're going to use this approach, you would need to ensure that the different groups contain participants with the same mix of ages and sexes and with the same amount of experience with the test. This experiment isn't an example of a reflex reaction because it isn't an automatic response, and it does involve the conscious part of the brain telling you we need to catch the ruler. Alternatively, we can test people's reaction times using a computer program in which each time an object appears on screen, the test subjects need to press a button as fast as they can. This allows higher resolution data to be collected, and it's more likely to give valid data as there's no way the computer can indicate it's about to make an object appear, even accidentally. However, in order to use this method, you need a computer and a program which might not be available for everyone, so if you're doing this in a class situation, it's probably not a good option. The endocrine system is the second system that's involved in homeostasis. Endocrine glands are cells that can secrete hormones. These are chemical messengers made of protein that are carried in your blood until they get to a target organ where the hormone needs to act. There are 6 specific endocrine glands in the body that you need to know the locations of. The pituitary gland, that master gland, is located in your brain. Your pancreas, which is part of your digestive system, is in the middle of your abdomen. Your thyroid gland is in your neck. Your 2 adrenal glands are just on top of your kidneys. And then if you're biologically female, you have ovaries, which are a little bit lower than that. And if you're male, you have testes, which hang outside the body. The pituitary gland is called the master gland because it's a gland that controls other glands. It secretes hormones into the blood, and those hormones will then act on these other glands to stimulate other hormones to be released. Your thyroid produces a hormone called thyroxine, and that stimulates your basal metabolic rate, which is necessary for growth and development. It's controlled by a negative feedback loop, so you make some, and then eventually when the levels in your body rise high enough, that's detected and your thyroid gland will stop making it. Your adrenal glands produce adrenaline, which is something that you make in times of fear or stress, and it leads to that fight or flight response. It increases the heart rate, and the point of that is to increase the amount of oxygen and glucose that your muscle cells and brain cells get, so that they can do more respiration and have more energy available for running away. It's important that your blood glucose levels remain constant, because without that you're not going to have regular energy levels, and also high or low blood glucose can cause damage to your cells. Your pancreas is responsible for both monitoring the blood glucose levels and for controlling them by releasing hormones. If your blood glucose gets too high, the pancreas makes a hormone called insulin, and this increases the permeability of your cells, particularly in the liver. That causes glucose to move into those cells, and that extra glucose can be converted into a polymer called glycogen for storage. If you're doing higher tier, you also need to know what happens when the blood glucose gets too low. The pancreas produces a second hormone called glucagon, and glucagon causes glycogen to be made back into glucose and released back into the blood. You can see the blood glucose level rises, and therefore the pancreas is stimulated. It produces and secretes insulin, which means releasing it into the bloodstream, and that insulin then stimulates glucose uptake from the blood, so the blood glucose level falls. If you're doing higher, you need to know that this will again stimulate the pancreas, and therefore glucagon is made and secreted. And glucagon will stimulate the liver to release glucose, so that glucose level rises. Linked to blood glucose, we can think about type 1 and type 2 diabetes. Type 1 diabetes is a disorder where the pancreas isn't making enough insulin, and so it's characterised by uncontrolled high blood glucose levels. You normally treat it with injections of insulin. It can be diagnosed with the 4 T's: going to the toilet all the time, being thirsty all the time, being tired all the time, and losing weight. Because that high blood glucose is really dangerous for the body, and the only way that your body can get rid of it is by putting it all into the urine. So you need to drink lots, and therefore you pee lots, and also because you're sort of throwing away that glucose rather than saving it for later, you're getting very tired and not having enough energy. Type 2 diabetes is a little bit different. It's not that the body isn't making insulin, but the cells are no longer responding to it. Generally, this can be controlled by controlling the amount of carbohydrates that you're eating, and also sometimes an exercise regime. It's known that obesity is a risk factor for type 2 diabetes, basically because if you constantly have high blood glucose levels because you're eating a lot of sugar, then it's likely that eventually your cells will become desensitized to that insulin, and they sort of start ignoring it. It's really important that the level of water in the blood is controlled, because if it isn't, then the red blood cells could either shrivel up or burst. We naturally take in water when we eat and drink, but we then lose some in an uncontrolled way when we breathe out the waste products of respiration, and also when we sweat, but also in a closely controlled way from our kidneys. In the kidneys, that water is used to dilute urea, which is toxic. This urea has come from the breakdown of proteins and their deamination from toxic ammonia. During puberty, the levels of reproductive hormones like estrogen and testosterone begin to rise, triggering secondary sexual characteristics, such as the development of breasts and facial hair. In females, the main hormone is estrogen, which is produced in the ovaries, and this is linked to the beginning of ovulation, where eggs start to mature and be released. Testosterone is the main male reproductive hormone, which is produced in the testes and stimulates sperm production. The menstrual cycle is controlled by 4 hormones. Follicle-stimulating hormone, or FSH, causes an egg to mature and become ready for ovulation. Then luteinizing hormone, or LH, stimulates that egg to actually be released. The ovaries are responsible for making estrogen and progesterone, and they're both involved in maintaining the uterus lining at the start of the menstrual cycle. At the start of the menstrual cycle, on day 1, as progesterone levels fall, the lining of the uterus is shed. FSH release causes an egg to mature and stimulates the production of estrogen. This causes the uterus lining to regrow, ready for a fertilised egg to implant. Estrogen inhibits FSH and promotes LH, leading to ovulation. High progesterone levels maintain the uterus lining, and if pregnancy occurs, progesterone levels will remain high. When you're picking contraceptive methods, you need to think about how reliable it is and therefore how likely it is you'll get pregnant, how easily available it is, whether there are any health implications or side effects, whether there are any ethical implications, and also how expensive it is. Hormonal contraceptive methods use the hormones produced by the ovaries to prevent the production of the hormones that are made by the pituitary gland. These are the ones that are responsible for eggs being mature and being released. Hormonal contraceptive methods include the pill, the coil, injections of progesterone, implants, and patches. If you're not using a hormonal method, which could be for all sorts of reasons, like the fact that some people are more susceptible to blood clots and estrogen can play a role in those blood clots, then there are non-hormonal methods that are available, such as condoms, which are what we call a barrier method, which is going to actually stop the sperm from getting to the egg, but also a diaphragm, and spermicidal creams, which you can use both with condoms and with diaphragms, and that spermicidal cream would actually kill the sperm. Some couples struggle to get pregnant and they may undergo fertility treatment. The first stage of fertility treatment involves taking a drug that contains FSH and LH. This is going to stimulate the maturity of ovules and also those being released, and so this can be done independently to increase the chance of a natural pregnancy, or it may be done alongside a drug that inhibits ovulation, so that maybe 20 eggs mature at once and can all be collected. The couple can then undergo in vitro fertilisation, which is where the sperm fertilises the egg in a petri dish. Once those eggs start to develop into embryos, when the cells start dividing, either one or several of them are transferred back into the mother's uterus. Although a fertility treatment does mean you have a better chance of becoming pregnant, it's obviously very emotionally and also physically stressful, and the success rates aren't that high. Particularly in older mothers, multiple embryos may be transferred to improve the chances of pregnancy, but this can then lead to multiple births, and it's more likely that you'll have twins, and that can be a risk both to the babies and to the mother. The second unit in Biology Paper 2 is Inheritance, Variation and Evolution, and the first thing we need to know about is the different types of reproduction. In sexual reproduction, we have gametes or sex cells. These are formed by meiosis. The gametes in animals are sperm and egg cells, and in plants they're pollen and egg cells. The nuclei from the gametes fuse in a process called fertilisation. This makes genetically unique offspring, even if both gametes come from the same parent. Don't forget, some organisms are able to self-fertilise and reproduce sexually with themselves. This is not the same thing as asexual reproduction. The offspring will be different to each other and different to the parents. Asexual reproduction by producing runners like a strawberry plant or spores like many fungi do doesn't involve gametes or fertilisation, and it produces clones which are genetically identical to each other and to their parents. You need to know about the two types of cell division. For paper 1 we focused on mitosis, which happens in all body cells and allows them to make more body cells for growth or repair. It starts with DNA doubling and then the cell divides once, so there are 2 diploid cells made. These cells are identical to each other and identical to the cell they were made from. Meiosis we've only really talked about for paper 2, and it happens in the reproductive organs. It's used to make gametes, sperm and eggs. It also starts with the DNA doubling, but then the cells divide twice. This means that instead of making 2 diploid cells with the same amount of DNA as the parent cells, 4 haploid cells are made with half the normal amount of DNA. The cells are different to each other and also different to the parent cell that they're made from. There is a lot of new vocabulary in topic 6 in GCSE Biology, and it's really worth making sure that you know the proper definitions, because this is one topic where a lot of students know what they're talking about but are unable to receive credit because they haven't used the right words. So firstly, gene and allele are commonly mixed up. A gene is a length of DNA that codes for a particular combination of amino acids to make a specific protein. So we could talk about a hair colour gene or an eye colour gene. The allele is then the variance of that gene. So for instance, a blonde hair allele of a hair colour gene. The genotype is about the actual alleles that you have in your DNA in your cells, and we're unable to tell this without genetic testing unless you have a specific phenotype. The phenotype is then what we can actually see, the expressed characteristics. So do you have brown eyes or blue eyes or blonde hair or brown hair? Expressed is a really important word. It means what can we actually see and detect. You should be aware that homozygous individuals have two of the same allele, and that could be homozygous recessive or homozygous dominant, whereas heterozygous individuals have two different alleles for a particular gene. Recessive means that the characteristic is only expressed in the homozygote. In other words, you would only know that an individual was carrying that allele if it had two of them. Whereas a dominant allele is expressed even in the heterozygote. So this would be something like brown hair, where even if you only have one allele, we still see that characteristic being expressed. The genetic material of a eukaryotic cell, like a human cell, is contained in the nucleus. This genetic information is made of a chemical called deoxyribonucleic acid, or DNA for short. DNA is an incredibly long molecule made of two connected chains that form a double helix, a sort of spiral staircase. These long pieces of DNA are wound up into little structures called chromosomes. When we talk about the genome of an organism, we mean the entire genetic material of that organism, the sequence of all the genes and also the intermediate pieces of DNA that don't code for proteins. The whole human genome has now been studied, and this will have great importance for medicine in the future. The Human Genome Project was a massive undertaking by scientists in many different countries. It ran from 1986, although it officially launched in 1990, up until its completion in 2003. The scientists sequenced 3 billion bases from 100 anonymous volunteers to allow them to search for genes linked to different types of disease, understand and treat inherited genetic disorders, and trace human migration patterns from the past. For genetics, it's quite likely that you get a question which asks you to either draw from scratch on the higher tier or fill in on the foundation tier a genetic diagram called a Punnett square. There are two genetic disorders that you need to know about in detail, so we'll use those as examples for how to fill in the diagram. The first disorder is cystic fibrosis. This is a disorder of the membranes where they produce lots of extra really sticky mucus, and so people with cystic fibrosis often find it difficult to breathe, or they have issues with their digestive system because they're swallowing that mucus and it's being produced in the digestive system and it's just getting in the way of digestion. When you fill in one of these Punnett squares, you're going to use a single letter to represent the alleles of this gene. And it's going to be a capital letter if it's the dominant allele and a lowercase letter if it's a recessive allele. For cystic fibrosis, we tend to use the letter F, but you're not going to lose marks if you pick a different letter if they haven't started it in the question. But the one piece of advice I would say is to use something where there's a clear difference between the capital letter and the lowercase letter. So A is great, B is great, C is a poor choice because if your writing is quite similar in size, your marker may not be able to differentiate between your capital and lowercase, and that makes it much harder for them to give you marks. So my first step in filling in this Punnett square might be I might need to identify what was going to go in these boxes up here that represent the gametes. Now in this question they have been given to me, but it's possible that in a higher tier question they might not be, and I might need to get that from the question. So remember that the two boxes at the top, they're coming from one parent, and the two boxes at the side are coming from a different parent, and it doesn't matter which way round you put them. So I can see that both of these parents are heterozygous because both of them have got both the uppercase letter representing the dominant allele and the lowercase letter representing the lowercase allele. So what I would then do is I would take these letters and I would add them into the squares so that everything that is below this capital F here is going to get a capital F, and everything that is below this lowercase f here is going to get a lowercase f, and then this capital is going to track across here, and this lowercase is going to track across here. And then the final thing that I need to do before I'm done with this question is identify which individuals have the disease. So cystic fibrosis is a recessive disorder, and that means that in order to actually have the disease, you need to have two recessive alleles. So this final individual here is going to have cystic fibrosis, but the other three will not. These two will be carriers, but they won't have the disease themselves, and this individual will be completely healthy. And I might be asked to express this in terms of a fraction or a percentage or a ratio. So just be careful. I've got 4 possible offspring here, and as you can see, 1 in 4 or 25% of those have the disorder. But watch out, if you're asked to give a ratio, my ratio here is 3 healthy individuals to 1 individual with the disease. It's not 4 to 1. The second example of a genetic disease you need to be aware of is polydactyly, and this is a disease where you get extra fingers or toes, and it's believed that Anne Boleyn had this. Now, this is an example of a dominant disorder, and this means that even if you have a single allele for polydactyly, you are going to be born with the disorder. Now, in this example, in this question, I've been given one parent who is heterozygous and one parent who is homozygous. But I'm going to follow that exact same process. So I'm going to track down from the top. This capital P goes into the boxes below it, and this lowercase p goes into the boxes below it, and then across from the left and across from the right. And then again, I'm going to identify these are the individuals that have got the disease and say, okay, that's 50%, or it's 2 out of 4, or however I'm asked to express it. Human sex is determined by 1 pair of chromosomes out of the 23 pairs that you have, and these are called the X and the Y chromosomes. If you're looking at a picture of the chromosomes, the X chromosome is a large, full-sized chromosome, whereas the Y chromosome is really, really small. Over thousands of years, it shrunk and shrunk until it's really quite tiny. The vast majority of people who are male will have 1 X chromosome and 1 Y chromosome, whereas people who are female will usually have 2 X chromosomes. That means that the sex of the baby is determined entirely by its father. The woman is always passing on an X chromosome in the egg. This means that 50% of babies are male and 50% are female, if we look at the whole population of the world. In one family, it might not be exactly 50-50. We can show how this works using a Punnett square like this. Embryo screening is a technique used by doctors with prospective parents who are worried that their baby is going to have a particular genetic disease that runs in one of their families. It can be used to look at the very early stage of pregnancy, often when the embryo is formed in a petri dish from IVF, to see whether the embryo has this particular genetic disorder that they're worried about. The point of doing this is to make sure that we're not bringing babies into the world who are going to really suffer from a disease. Also, often the cost of treating these disorders and trying to keep a baby with the disorder alive can be really high, so there is an economic component as well. Often when this is done, the parents will undergo IVF treatment and produce a lot of embryos. Leftover healthy embryos can be donated for scientific research or for treatment like looking for stem cells. There are some downsides though. Obviously, there are some pretty big ethical issues. If you're producing a lot of embryos, the ones that have the disorder are going to be destroyed and rejected, but also even some healthy embryos may not even be transferred or attempted to implanted. Some people are okay with that because we're just talking about a tiny bundle of cells that can't feel pain, but some people believe that life begins at conception and would not be okay with this. There's also the potential, albeit not in the UK at the moment, for people to have designer babies. So some people are worried that parents would want to screen their babies for, say, blue eyes. In the UK, you can only even screen for sex if your family has a disease that is really strongly linked to the sex chromosomes. So say it might be easier to identify male embryos and discard them than actually screen for the particular gene on the sex chromosomes that's causing a disease. There's also an implication here that the babies that would have the genetic disease are undesirable, and there's a risk that by using embryo screening, we're feeding into an idea that people born with the disease are somehow less good, and that could lead to prejudice. Also, the embryo can't consent. It's just a ball of cells, so there's another ethical issue too. Embryo screening tends to be quite an expensive process, and if you're doing the version where you screen the baby inside the mother's uterus, then there's a risk of miscarriage. There's also a chance of false positives, so you could tell a woman that the baby has a genetic disease, and she could decide to terminate that pregnancy, but there's actually a tiny but non-zero chance that you're wrong, and she's now terminated a healthy pregnancy. This topic is a perfect example of where you might encounter an evaluate question. If you do, remember it's vital that you compare both sides of the story, in this instance giving both advantages and disadvantages. Make sure that you use data if they provide it, and you have to write a strongly justified conclusion. Variation is differences between individuals of the same species, and it can have a genetic root, an environmental root, or be a combination of the two. Genetic variation arises due to natural mutation, and it can be passed on if that mutation is happening to cells that are producing gametes. But most mutation has no effect on the phenotype, either because it doesn't actually change the amino acid sequence of a protein, or because it's happening in a non-coding region, or because that mutation is not happening in cells that are responsible for producing sex cells.
In GCSE biology, the term evolution refers to a change in inherited characteristics over time as a result of natural selection, and it may result in the formation of a new species if two populations of one species end up becoming so different to each other because of evolution that they're no longer able to interbreed and produce fertile offspring. We now believe that all species evolved from simple life forms that first developed more than 3 billion years ago.
In the exam, it's quite likely that you could be given some information and then asked to explain how a particular characteristic has evolved. Natural selection begins with changes in the genome brought about either by mutation or by sexual reproduction, leading to novel combinations of alleles. This means that individuals of a particular species will show variation for a particular characteristic. A selection pressure can then act on this. This might be the likelihood of being killed by a predator or access to a particular food source. The individuals that are best adapted to that environment are most likely to survive and, crucially, to breed successfully. In doing so, they're able to pass on their alleles, and therefore the next generation of that species will have more of that particular successful characteristic. For instance, pre-industrial revolution, white peppered moths were well camouflaged on silver birch trees, and on the rare occasion that mutation led to a black moth to be born, it was quickly seen and eaten by a predator. However, when the factories started producing lots of black smoke and the trees gradually became darker, the darker moths had an advantage. They were able to survive for longer because they were camouflaged and to breed and pass on their alleles. Within just a few years, there were far more darker moths because the dark moths had been able to have more babies. However, when the Industrial Revolution finished and the trees reverted to their original colour, so did the moths. Another really good example of evolution by natural selection is the evolution of antibiotic-resistant bacteria. Bacteria are able to evolve faster than multicellular organisms because they have short generation times and a higher rate of mutation, and additionally, because they only have one copy of each gene, any mutation is much more likely to affect their phenotype, so evolution can actually act on it. Antibiotics are molecules made by fungi or synthetically in a lab, which can be used as drugs to kill bacteria, although not viruses. Normal bacteria are susceptible to antibiotics, which means if you take the drug, then quickly all the bacteria are killed. However, some bacteria mutate and become resistant to the antibiotic. When the drug is used, the susceptible bacteria die, but the resistant ones live on. Some people, because they feel better, stop taking the antibiotic. Now, free from competition, the resistant bacteria can reproduce. If the patient had taken all of the antibiotics, then all the bacteria would have died eventually. There are a number of things that we can do in order to prevent the increase in the number of antibiotic-resistant bacteria. The first one is to not overuse antibiotics for infections that will clear themselves up on their own, or for infections that are actually viral and wouldn't be affected by antibiotics anyway. The second really important one is to make sure that you are actually finishing a course of antibiotics so that all the bacteria do die. We try to make sure that we're using different antibiotics for animals compared to humans, and if we can do, we only use specific antibiotics which only work on a particular bacterium. It's important to maintain hygiene and avoid spreading bacterial infections around, and if somebody does have an infection with an antibiotic-resistant bacteria, they're kept in isolation to prevent them passing it on to anybody else. Selective breeding, which we sometimes call artificial selection, is the process of breeding plants or animals for particular genetic characteristics. It's how farmers make evolution happen really quickly so that they can get a new crop or a new herd of animals which have the best characteristics. These are going to make them the most money. The first point is that we need to start with a population with some variation in it. And from that population, we're going to pick the individuals with the best characteristics so that we can breed them together. This could be cows producing lots of milk, or pigs producing lots of meat, or sheep producing lots of wool. So you're going to pick a really good male and a really good female and breed them together so that they have lots of offspring. And then out of those offspring, you're going to pick the ones that have the desired characteristic, because with sexual reproduction, we do have variation in the offspring. And then you take those offspring and you breed those together, and then you look at their offspring and see which ones of those have the desired characteristic. and you're going to repeat this process over several generations until you have a population that all have the characteristic that you're trying to get. Genetic engineering, or genetic modification, is an even faster and more efficient way to develop a population that have a particular characteristic. So rather than taking two parents who have the characteristic and hoping that their offspring will inherit it when they're bred together, we can actually take the gene and directly insert it into a new individual. And this also makes it possible to transfer genes from completely different species. Plants have been genetically engineered to make bigger fruits, but also to be resistant to different diseases where that particular species isn't currently resistant at all. It's also been used for things like genetically engineering bacteria to make human insulin, and this is really important for treating diabetics who need a constant supply of insulin. If you're taking higher tier, you need to be able to describe how this process is done. Special enzymes called restriction enzymes are used to cut out the required gene from an organism that already has that gene. This piece of DNA is then inserted into a vector, like a bacterial plasmid. This is basically another piece of DNA that is going to make it easier to get the gene that we're interested in into the organism we're trying to transform. That vector, which could be a plasmid, is put into the organism we're actually interested in. And this is done at a very early stage, because if we do it when there's just a single cell, then when the organism grows up, every cell will be transformed with the new gene. Whereas if we waited until it was already an adult, then only part of the organism would be transformed. Despite being expensive and quite hard to do, GM technology has a major advantage in that it's extremely precise because we're only transferring specific genes that we already know the function of. However, it does have some drawbacks. One major use of GM is to make crops resistant to herbicides, which are chemicals that kill plants. This means that rather than carefully spraying herbicide just on the weeds, you can spray it on the entire field without killing your crop. However, if the crop then breeds with a weed to produce a hybrid, these could be resistant to the herbicide too. Another way GM is used is to make crops that contain cryprotein, which is toxic to insects that try to eat the plant. This prevents you needing to spray insecticide on the crops. However, some people are concerned that this could still have an impact on other insects like pollinators. Because GM technology is so new, there's been limited research into its impact on human health, which makes some people nervous about eating GM crops, even though we know the function of the small number of genes transferred. Modern medical research is exploring the possibility of genetic modification to overcome some inherited disorders. When we say that a species has gone extinct, like the western black rhino in 2011, we mean every single individual of that particular species has died out. There are no more, and they're not coming back. This can happen because of a new disease, a new predator, and here we do include humans in hunting, a new competitor that's better at getting food, a natural disaster like volcanoes, temperature change, which is particularly applicable if you're thinking about plants and how they're maybe unable to move range, or insects that are not able to move their range as quickly as the plants that they feed on or pollinate, and also something called the cyclical nature of speciation, which basically means that over very long periods of time, one species ended up turning into a completely new species, and so the first species has effectively gone extinct because there are none of that old type of organism left. When we're trying to learn about extinct species, the fossil record is really important and really useful for studying evolution, because we know that the deeper down in rock we find a fossil, the older that rock probably is, and therefore the longer ago that species was alive. When we say fossils, we mean the remains of an organism that have been preserved, usually in rock, but sometimes in ice or in amber, and usually over millions of years. Most of the fossils that we have are the rocky type, and they form when an organism dies and then layers of sediment, so sand and mud, have formed on top of the remains, and that's usually going to happen under the sea. Because oxygen isn't able to get in, bacteria aren't able to decay things. So over a very long time, the hard parts like the bones or the teeth can become mineralized. So minerals take the place of the bones. And basically we end up with a rock that has the shape of the bone or whatever it is inside it. And this kind of explains why we don't really have many fossils of things like jellyfish or worms, because they didn't have hard parts to fossilize. It's also possible to fossilise other indications that an organism was around other than the organism itself, so that could be prints or burrows or even its droppings. Classification is the science of putting things into groups based on their shared characteristics, and you need to know about two systems: the old school taxonomic hierarchy system and also the three-domain system as proposed by Wurzer in 1990. In the three-domain system we have eukaryotes, including animals, plants and fungi. then the true bacteria, and finally the archaea, which are as different from the true bacteria as they are from us. But according to AQA, they're primitive bacteria usually found in extreme environments. In the traditional system, we had 5 kingdoms: animals, plants, fungi, bacteria, and Protoctista, which contains algae and slime moulds, and also Plasmodium, which causes malaria. Each one of those kingdoms is split into a number of phyla. So for instance, our phylum as humans is called the chordates, and that includes everything that has a spine and a backbone, as well as a few other things that don't. Then we split that down further, so we have our class, and our class as humans is mammals. So that can be defined as organisms that produce milk and have body hair and give birth to live young. Then we split further into orders, and ours is primates, which includes all the sort of monkeys and chimpanzees and things like that. And then we split into families, and ours is hominids, so things that are kind of human-shaped basically. And finally we have a genus and a species, and these together make up what's called the binomial name. Sometimes people call this a Latin name, but actually loads and loads of these names are not Latin, so you shouldn't call it that, and also you won't get the mark in the exam. It's called the binomial name, with a binomial system. So every species has a binomial name which has 2 parts. And the first part is the genus, and the second part is the species. So for instance, you'll have heard humans called Homo sapiens. Homo is our genus, and then there are some other species that are part of the Homo genus, and then sapiens is our species name. An ecosystem is the interaction of a community of living organisms, in other words the biotic factors, with the non-living or abiotic parts of their environment. In order to survive and reproduce, organisms require a supply of materials from their surroundings and also from the other living organisms there. Animals and plants may compete within their species or intraspecifically, or between species, i.e. interspecifically, for both the biotic and the abiotic factors. Where an organism is able to live and survive and thrive is going to be based on a combination of biotic factors and abiotic factors. The biotic factors could be things like the availability of food, so either prey if you're a carnivore or plants and fruits if you're a herbivore. It'll be based on predators which are going to try and eat you, competitors for your food and also for things like shelter and mates, whether there are diseases, and also how easy it is for you to find a mate. It's no good you finding the perfect environment where you can survive for ages if you can't actually have any babies there. And then the abiotic factors, for plants, light intensity is obviously going to be a big one so they can photosynthesize. The temperature, the moisture levels, the mineral ions, the soil pH, and also carbon dioxide levels. And then if you're an aquatic organism like a fish or a newt, then oxygen levels in the water are also going to be really important. When we talk about interdependence, we're talking about how species depend on one another for survival. So for instance, maybe insect A drinks the nectar of plant C as it's pollinating its flowers, and mammal B eats its fruit. And therefore plant C is dependent on insect A to pollinate it, and also mammal B to disperse its seeds, which are inside the fruit after it's eaten them. This is called interdependence, and in a stable community, all of the species and the environmental factors are in balance, so that the population sizes remain fairly constant year to year. Over time, species become adapted to the environments they're living in, and here we're talking in evolutionary timescales. So we're not saying that one bear grows a thicker coat because the winter is cold. We're saying that over thousands of years, the bears with the thickest coats survive best and pass on their alleles. So bears as a whole develop thicker coats. You may be asked to describe how animals and plants are adapted to different environments, such as how plants growing in the desert have small leaves and few stomata to avoid water loss, as well as large root systems to absorb maximum water when it rains, or how animals living in the Arctic have white fur for camouflage and to keep them warm. Feeding relationships in an ecosystem can be represented by food chains, which can be put together to make a food web like this one. In each food chain, the direction of energy transfer is shown by the arrows, which always go from the organism being eaten to the organism that is eating it. Each food chain begins with an organism that can synthesize molecules containing energy, and we call these organisms producers. For the purposes of GCSE, producers are green plants or algae making glucose by photosynthesis. Producers are then eaten by primary consumers, which we can also call herbivores, and then primary consumers are eaten by secondary consumers, and secondary consumers by tertiary consumers. We can also say that anything that kills and eats other animals is a predator, and anything being killed and eaten is prey. In a stable community, the numbers of predators and prey rise and fall in cycles. The ecology required practical centres around measuring the population size of a common species such as clover, usually in response to an abiotic factor like light, moisture content or pH. To estimate the population size, you use this piece of equipment, which is called a quadrat. It's a square frame, and it often has divisions inside to help you estimate what percentage of the quadrat contains the plant you're sampling. Depending on your exact experiment, you may be comparing two separate locations or looking at a transect, which is a long, straight line where an abiotic factor changes gradually. If you're comparing two locations, you use random sampling to ensure you have a fair representation of each location. Split the area into a grid using coordinates, and then use a random number generator to tell you which squares to sample, for instance square B1 and C5. At each location, you sample the coverage using the quadrat, and also take a reading of the abiotic factor, such as a light reading with a light meter or measuring acidity with a pH probe. If you're looking at a factor that gradually changes, such as moisture content as you get closer to a river, then you can use systematic sampling. You use a long tape measure to make a line called a transect, and then put down the quadrat at regular intervals along the way, say every 5 meters. At each location, you survey percentage cover and your abiotic factor. There are some opportunities in here to test your math skills, and they usually involve working out the area of the sample site or the quadrat surveyed. These are usually quite easy to do, but also easy to make silly mistakes with, so make sure you check your working. Materials are cycled through ecosystems as part of both biotic and abiotic processes. You could be asked about the water cycle, which provides fresh water for animals and plants. The sun's energy causes large bodies of water, such as lakes, to evaporate. This leads there to be water vapour in the atmosphere, which may then condense to form rain clouds. These are responsible for precipitation or raining, so that water falls down onto the ground. Drainage then leads this to go back to lakes, but in the meantime some of it is intercepted by plants, which release it again via transpiration. You could also be asked about the carbon cycle, which contains biotic and abiotic processes. The total amount of carbon in the atmosphere in the form of CO2 is increased by respiration from animals, plants and microorganisms as they break down glucose and release carbon dioxide. It's also released by decomposition, in which microorganisms break down dead plants and animals and then release carbon dioxide into the atmosphere as they respire, as well as mineral ions into the soil. Carbon dioxide levels are also increased abiotically by the combustion of fossil fuels. Meanwhile, carbon dioxide is removed from the atmosphere by photosynthesis, in which green plants and algae absorb carbon dioxide and make glucose. They then store this as starch and use it to make fats, proteins and cellulose. Carbon dioxide is also removed when it dissolves in oceans and forming into carbonates, which are locked up in sedimentary rocks, and by the production of fossil fuels in peat. Carbon is also transferred between plants and animals when animals eat plants. Biodiversity is all about having a lot of different species either on Earth or in a particular ecosystem, and the reason we care is that the higher biodiversity is, the harder it is for that ecosystem to crash. Basically, if you have a system where you maybe only have 2 or 3 species and they're completely dependent on each other, if something bad happens to one of them, then everything else comes crashing down. Whereas if you have lots of different species and maybe everything has 5 or 6 different sources of food, then even if something bad happens to one population, everything else is able to stabilise and recover. There are lots of different actions that we as humans can take to protect and encourage biodiversity, including things like breeding programs for endangered species, protecting and regenerating rare habitats, and trying to reintroduce field margins and hedgerows around farms, because just having a monoculture is not great for biodiversity. There's also reducing deforestation, recycling resources, and also conserving peat bogs. In terms of other things that humans are doing, there's obviously a lot of pollution being caused, which kills animals and plants and reduces biodiversity, in terms of water and in air and on land. Us building, quarrying, farming and dumping waste all contribute to us reducing the amount of land and habitat available to animals and plants. And also we've carried out large-scale deforestation, particularly in tropical areas such as the Amazon rainforest, both to provide land for farming, for cattle and rice, and also to grow crops for biofuels. Carbon dioxide, methane and water vapour are all examples of greenhouse gases, which are molecules that trap infrared radiation, causing Earth to warm up. Having a little bit of them is a good thing because it keeps our temperature stable and high enough to sustain life. But unfortunately, due to human activity, we now have too much of them, and so climate change and global warming are happening. These greenhouse gases form a blanket around the Earth, through which small wavelength radiation, like visible light, is easily able to pass. However, when that radiation reaches Earth, it's absorbed, and when it's re-emitted, it's done with a slightly longer wavelength, as infrared radiation. this infrared radiation is too large to pass through that greenhouse gas blanket. And therefore, although some of it does escape, a lot of it hangs around and causes the Earth's atmosphere and ultimately the Earth to warm up. This leads to rising temperatures, which lead to some pretty serious consequences. As the ice caps melt, this causes sea levels to rise, and this can cause flooding and other extreme weather events such as drought and hurricanes. Polar habitats are being lost, and with them many species are going extinct. Try to be as specific as you can while describing the consequences. Talk about polar or Arctic habitats and name specific extreme weather. Thank you very much for watching, and I hope that you're now feeling slightly better prepared for your exams. If you have found this useful, then don't forget to like and subscribe and interact with the video, and I'll bring you some more GCSE Science revision videos soon.